Testcross, a genetic tool. Testcross is a genetic technique used to determine the genotype of an individual with a dominant phenotype. It involves crossing an individual with a recessive homozygous individual. The resulting offspring can reveal whether the dominant phenotype individual is homozygous dominant or heterozygous. How does it work? Dominant phenotype. An individual with a dominant phenotype could have either a homozygous dominant, AA, or a heterozygous, AA, genotype. Recessive homozygote. The individual used for the Testcross must have a recessive homozygous genotype, AA. Offspring analysis. If all offspring have the dominant phenotype, the original individual is likely homozygous dominant, AA. If some offspring have the recessive phenotype, the original individual is heterozygous, AA. Example 1. Determining the genotype of a purple flower. Suppose you have a purple-flowered pea plant. You want to determine if it is homozygous dominant, PP, or heterozygous, PP. You cross it with a white-flowered pea plant, PP. If all offspring are purple, the original purple-flowered plant is PP. If some offspring are white, the original purple-flowered plant is PP. Example 2. A real-world application. In agriculture, test crosses are used to determine the genotypes of plants with desirable traits. For example, if a farmer has a high-yielding corn plant, they can cross it with a recessive homozygous corn plant to see if the high-yielding trait is dominant or recessive. This information can be used to breed for even higher-yielding corn varieties.